to translation. So it's sort of the last bit of information regarding the different cellular processes that are utilized involving genetic information. We have central dogma, that's replication, transcription, translation. This particular lecture is really going to focus on the mechanism of translation. So this is a process where the genetic message encoded in mRNA is translated on the ribosome into a protein. So just a quick introduction to protein metabolism. So a typical cell is going to require thousands of different proteins at any given moment. And those proteins, they need to be targeted to their appropriate cellular location. And once they're there, they become degraded when they're no longer needed. Protein synthesis itself is the most complex biosynthetic process. So it requires 30 different macromolecules, so those are going to be proteins and RNAs that are going to be involved in making a polypeptide. And protein synthesis itself is a very energy consuming process. So up to 90% of the chemical energy used by a cell is going to be involved or is used by protein synthesis. So it requires a lot of energy. And despite that, despite how much energy it takes, despite how complex it is, protein synthesis actually occurs very fast. So a polypeptide of 100 amino acid residues can be synthesized in an E. coli cell at 37 degrees in 5 seconds. That's pretty amazing if you think about it. And we're going to go through that process, and you'll see how complex it truly is. So let's kind of talk about the genetic code and what codons are. So the amino acid sequence of a protein is really going to be defined by the continuous triplet bases in an mRNA sequence. And these triplet of bases, each triplet represents a codon. So a codon is a triplet of nucleotides that's going to then code for a specific amino acid. During translation, these triplets in the mRNA, they're going to be read in sort of this successive, non-overlapping fashion that's written from 5' to 3'. So the mRNA is going to be read sort of a triplet of base at a time, again going from the 5' to 3' direction. And the first codon that's in the mRNA sequence is going to establish the reading frame, in which a new codon is going to begin every 3 nucleotide residues. And so if you have an insertion, so addition of a nucleotide or deletion of a nucleotide, that's going to cause a frame shift. So it's going to shift this reading frame, and it's going to cause a mutation, which often will which normally changes the sequence of the protein that's synthesized, and often it's going to cause a truncated protein that is non-functional. So shown here is a codon table, and you can kind of think of a codon table as a dictionary of amino acid code words in mRNA, where each set of 3 letters again represents an amino acid. So there are 64 possible codons, and 61 of these codons are going to actually code for a specific amino acid. AUG, so this is AUG here, is the initiation codon or a start codon. So it's going to be the signal for the beginning of a protein or polypeptide. And then AUG can also code for methionine residues that are in internal positions of polypeptide. All of these amino acids, except methionine and tryptophan, they have more than one codon. And in most cases, the codons that specify the same amino acid actually only differ at the third base. So for example, if you look at threonine here, you can see that the first and second base in the codon is the same AC, but it's only different by UCAG between these 4 different codons that all code for threonine. And then we have termination codons, so that's UAA, UAG, and UGA. So that's going to essentially signal for the end of translation, end of synthesizing a polypeptide chain. If you have a mutation that causes, that converts a codon into a stop codon, again, this is going to cause a truncated gene product, which will normally result in again, a truncated protein that's non-functional. And so when that happens, we call this a nonsensical mutation. So the genetic code is degenerate or redundant. To mean degenerate or redundant means that amino acid may be specified more than one codon. So you can see that here's the different amino acids, and most of these amino acids, they have more than one codon that is tripleted bases that's going to specify for that residue. And the only exception is this is, again, methionine and tryptophan. It's only specified by one specific codon. And then, although an amino acid may have one or two more codons, each codon specifies only one amino acid. So the genetic code is nearly universal. So that means these codons that specify the amino acids, it's going to be the same for whether it's humans, E. coli, plants, amphibians, and yeast. They all share the same genetic code. There are some exceptions, and that's seen in mitochondria and some archaea bacteria. Okay, now even though the codon usage is, the codon itself, or the genetic code is universal, there is some what's called codon bias or codon usage. So not all codons in a given amino acids are equally prevalent. Different organisms actually differ in the codon usage. So that is, they actually use a specific set of codons that are used to code for a specific amino acid. So for example, let's just take alanine. So alanine is coded by these 4 codons, GCU, GCC, GCA, GCG. And if we compare that usage of that codon between E. coli and humans, they're actually quite different. You can see that in E. coli, GCG is much more frequently used, whereas in humans, this GCC is much more frequently used. And so this is what we call codon bias. There is a certain propensity for the usage of a codon in that given organism. And because of that, sometimes that can create an inefficiency when an organism tries to translate a gene from another organism. And so if you are doing recombinant protein expression, meaning you're trying to express a gene from one organism in a different type of organism. For example, we do this all the time in the lab. We take human genes and try to express it in E. coli. Then you can often have problems because, those tRNAs that are in the E. coli may end up actually be limiting if you're trying to express a human gene. So this is why expressing a human protein in E. coli is not always successful. Oftentimes, you may have translation problems which may then lead to folding problems. You may not be able to translate or make a protein at all. And actually, some E. coli have been engineered to express a different set of tRNAs that have specificity for what we call these rare codons, so codons that are more common in humans. So those are for you guys that are involved in protein expression in E. coli. Okay, so let's talk about codons and anticodons and tRNA structure and function. So transfer RNAs or tRNAs, this is sort of a diagram of their structure. They are going to base pair with codons through these anticodons. So it's a 3-base sequence that's located sort of in this bottom stem of the tRNA. And so the first base of the codon is going to base pair in the normal Watson-Crick, usually normal Watson-Crick base pairing with the third base of the anticodon of the tRNA. The second base of the codon will base pair with the second base of the anticodon of the tRNA, and then the third base of the codon will base pair with the first base of the anticodon of the tRNA. And just like in DNA or DNA replication, or whenever you make any type of DNA-RNA hybrid, or any really type of double base pairing or base pairing of a nucleotide, the alignment of the two RNAs, that is the tRNA and the mRNA, they are anti-parallel. So the tRNA is going from 5' to 3' in this direction, and then the mRNA is going from 5' to 3' in the opposite direction. And then the first two letters of the codon, so here it would be A and U, is going to be the primary determinants of specificity. So this third codon, which is shown as C, that can usually be different, and it will still end up coding the correct amino acid. And that's actually quite important in terms of helping to increase the speed of translation. So there is something called the wobble hypothesis, meaning that the first two codons of the mRNA is going to form a very strong Watson-Crick base pair with its corresponding base in the tRNA anticodon. And again, that's going to confer most of that specificity of coding the right amino acid. However, that third base, which we call the wobble base, it does not necessarily have to have this perfect Watson-Crick base pairing. In fact, it can wobble, meaning it can be a different pace other than a normal Watson-Crick base pairing. And so this is actually really quite important because it seems that only having two strong base pairings, that first and second codon, is important, and that having the third one wobble actually helps the tRNA sort of dissociate faster from the translational machinery and help to speed up translation. Yes, Pat?
So is this effect caused by the geometry of the tRNA strand, or what makes this? Is the third base farther away? Is that why it doesn't base pair as strongly, or how does that work?
That I'm not entirely sure what exactly is going on at the molecular level that allows other bases to be accepted in that third position, other than the fact that you just need to have a good, strong hydrogen bonding for the first and second position. So I don't know. Yes, Mitch, do you know?
I was just reading it's rounded at the bottom, right? At least somewhat rounded. So could it be that it's tilted a little bit?
I don't know. I'm not sure. I'd have to really take a look like very, very closely at to the structure of what exactly is going on there. And I don't think I have a structure of what that actually looks like in the ribosome. So great question. Okay. That's a really great question, but I don't know the answer to this. We're going to actually skip this slide and go on to the next slide. Okay, so it's interesting that we have this, that we can have kind of this wobbling effect of the third nucleotide. Meaning, you can have different nucleotides at that position, at that third position, and still encode for the correct amino acid. Now it turns out that the genetic code is what we call mutation resistant. The types of damage that's usually caused in the genome involves what's called a transition mutation. And that means that if a purine something happened, like you have a lesion. So, and let's say it's a purine, so like an adenine. So if you have something that happens and you need to replace it, then usually that replacement is still going to be a purine. So so if you have like an adenine, it'll usually be changed to a guanine. If you have a thymine, it's usually going to be that something happened to the thymine. It would usually be changed to a cytosine. So a purine would be replaced by a purine, and a pyrimidine is replaced by a pyrimidine. So that would change a GC base pair to an AT base pair if it was something wrong with the G. So each position in a codon has actually evolved so that there's actually some resistance to these types of mutations. Now, probably the best mutation that can happen is a silent mutation. And so a silent mutation is a mutation in which there's a change at that third position of a codon, and that would cause a change in the DNA sequence, but it does not change the sequence of the protein that it would code for, because you would still end up getting the same amino acid at that codon position, without changing the amino acid sequence. So that would be a silent mutation. If a mutation changes the code to a different amino acid, this is a missense mutation. So typically, if you have a change in the first or second position of a codon, that's going to change the actual amino acid. Now, it's interesting because you would think that this would be very deleterious. It could, really mess up the structure and the function of a protein. But again, the genetic code has sort of evolved to be able to handle some of these mutations that can cause a change in the sequence of the protein. So for example, if you have a mutation in the first position of a codon, it may be deleterious, or it may not be, because often that changed amino acid actually has similar properties. So for example, GUU, that codes for valine. If there was some type of mutation that causes the G, a purine, to be replaced by another purine, then that would give you AUU, and that's isoleucine. And actually, the chemistry of valine and isoleucine are really quite similar, right? What's similar about those two amino acids? Yeah, they're both hydrophobic and they're both branched chain. So maybe that when the protein actually folds into its final structure, that it may be able to sort of tolerate an isoleucine versus a valine, because it's really not that different. Okay, so I don't know. I just think that's pretty cool. So now we're going to go over protein synthesis, and like I said, it's a very, very complex pathways, and it involves initiation, elongation, and termination steps, just like we saw in replication and transcription. Although, again, it's so much more complex compared to transcription and replication. And then there's also additional stages, so there's going to be a part where the amino acid needs to be activated and attached to a tRNA. And then there's also this post-synthetic processing of the complete polymer, essentially post-translational regulation of the protein. So let's first talk about the secondary structure of tRNA. So this is kind of showing that base pairing of the secondary structure. So tRNA are sort of the adapters for translating the language that's written in our DNA of a nucleic acid into the language of a protein as functional product. So you have hydrogen bonding in the actual tRNA structure, or in the actual tRNA sequence, and that's going to form sort of this cloverleaf structure that has 4 hairpin arms. So you have a tau psi C arm, you have an anticodon arm, you have this other hairpin called the D arm, and then you have this amino acid arm. So at the end of this anticodon arm, you have the anticodon loop, which then is going to contain your anticodon. So those are unpaired nucleotides, and the anticodon sequence is going to base pair with the codon. You have at the 3' end of the transfer RNA this sequence of CCA, and so this adenine at that 3' end is going to be the point of amino acid attachment. And then here's the 3-dimensional structure of a tRNA. It has sort of this twisted L conformation, and here you have your amino acid arm your amino acid arm on the bottom, or sorry, up here at the top, and that's at the opposite end of the anticodon arm at the bottom. And then you have your 2 other arms that make up the middle. And then this tau psi C arm is the portion of the tRNA that is going to actually interact with the large subunit of the RNA. So first we're going to talk about the actual process of attaching the amino acid to the tRNA, also known as amino acid activation. So you have amino acids, and then you have these specific tRNAs, and you always have to make sure that the correct amino acid is attached to its cognate or specific tRNA. And the importance of doing this attachment of actually covalently attaching the amino acid to the tRNA is it'll actually activate the carboxyl group, so that way when we actually get to the point of doing protein translation, of making polypeptide, it's going to help to facilitate the form of a peptide bond during protein synthesis. It's going to also establish this link between the amino acid, so you have to somehow link the amino acid to the information that's going to be encoded into the mRNA molecule. And so the tRNA is going to be that link. And so aminoacyl tRNA synthetases, these are the enzymes that are going to attach the correct amino acid to their tRNAs. It takes place in the cytosol. And so there are specific aminoacyl tRNA synthetases that are going to attach the amino acids to their corresponding tRNAs. And these enzymes are actually highly specific. So they're very specific in terms of their amino acid and the tRNA. So most organisms actually have one aminoacyl tRNA for each amino acid. And then for amino acids in which there are 2 or more corresponding tRNAs, it's going to be the same enzyme involved in doing the amino acylation reaction. So this is the actual reaction that's catalyzed. It's essentially a 2-step reaction, catalyzed again by the amino tRNA synthetase. So in this reaction, the amino acid is going to form a covalent bond. It's going to react with ATP, and it's going to form a covalent bond with the amino acid and the ATP. So they'll form an aminoacyl adenylate or aminoacyl AMP, and then displace pyrophosphate. And so the hydrolysis of ATP is going to provide the energy for that bond formation, as well as the hydrolysis of the pyrophosphate into 2 inorganic phosphate. And then in the second step, the aminoacyl moiety that is now activated because it's attached to the AMP is going to then react with tRNA, and then the aminoacyl group will be then attached to the tRNA, and AMP is displaced to give this final reaction, which is the amino acid plus ATP plus tRNA. tRNA gives the aminoacyl tRNA, AMP, and pyrophosphate, which again, the pyrophosphate will get hydrolyzed to 2 inorganic phosphate. So here's a specific mechanism of that reaction. The first is going to be the activation of the amino acid by adenylating it. So here's your amino acid. This is your alpha carbon, R group, alpha amino group, and then the alpha carboxyl group. The carboxyl group is going to attack the alpha phosphate of ATP and displace the pyrophosphate group, resulting in an adenylated amino acid. So this is a aminoacyl adenylate intermediate. And then this intermediate can go down one of 2 different pathways, depending on whether it's a class 1 tRNA synthetase or a class 2 tRNA synthetase. So if it's a class 1 tRNA synthetase, what will happen is that amino acid will be attached to first to the 2' hydroxyl group of the ribose on that 3' adenine of the tRNA, and then transferred to the 3' hydroxyl group. And so what this looks like is, here's your aminoacylated molecule. Here is the tRNA, and remember we have at the 3' end that adenine that is going to then be the site of the amino acid attachment. So if it's a class 1 tRNA synthetase, that 2' hydroxyl group will attack the carboxyl group of the aminoacyl AMP, and then displace the AMP moiety, giving us this tRNA molecule where now the amino acid is attached to the 2' group of the ribose ring. In the next reaction, we essentially have an isomerization reaction where the 3' hydroxyl group will attack the carboxyl group of the amino acid, displacing this oxygen at that 2' carbon of the ribose. So now the amino acid has been attached to the 3' carbon of that adenine at that 3' end of the tRNA. So that's a class 1 synthetase. Class 2 involves direct attachment of the amino acid to that 3' hydroxyl group of the adenine of the ribose ring. So here's a 3' hydroxyl group. It'll attack that carboxyl of the amino acid, displacing AMP, and now we finally have the amino acid attached to the 3' carbon of the ribose of the adenine. So I don't know. I always just think it's amazing. You have essentially and tRNAs are so cool because once they've been activated, you have essentially an RNA amino acid hybrid. I mean, it's just the coolest thing. Yes, Pat.
So where did the adenine that's already attached to tRNA?
Where did so that was coming from just the cellular ATP that's around. Okay. So the amino acid first reacted with the ATP, so that actually creates a carboxyphosphate that activates the molecule. So, and that's our aminoacyl adenylate molecule, and that actually activates this this for this second reaction. But again, that adenine or the adenylate actually just came from ATP, and again, ATP is, there's just actually lots of it in the cell.
But then there's another
Adenine?
already on the tRNA.
Yes, so great question. So if you remember, yes, let's go back to, let's see, what's the good, okay, so that adenine, again, that point of attachment of the amino acid was already there in the tRNA molecule. And
It's just already there.
it's already there. And in fact, I'm going to just show you guys something really quick. I'm going to show you this to you guys. Now, I'm not going to test you guys on this, but if you guys remember, let's go back to transcription. Now, I said I wasn't going to test you guys on post-transcriptional modification of tRNAs, but if you remember that one of the modifications that occur is once the tRNA is synthesized, once it's been transcribed, there's a lot of different modifications that happen. One of these is that there's actually cleavage of the 5' and 3' end, and then you have this addition of the CCA at the 3' end. And so that's where that actual adenine comes from, is through post-transcriptional modification. Where were we? Okay. Does anyone have any questions, any other questions about the chemistry of the tRNA synthetase reaction? Yes, Mitch? No? Okay. I always like to ask a question about this reaction on an exam, just because I like it.
Actually,
Yes?
is there a benefit of the class 1 over the class 2? Because the class 2 just attaches directly to the 3'.
You know, that's a great question. The class 1 seems to be specific for specific amino acids, and the class 2 has its own amino acids that's specific for. And I mean, I can't remember what they're exactly which ones are specific for what. And I don't know what the benefit would be of why you would first attach it. So here, this makes more sense because it makes more sense to directly attach it to the 3' hydroxyl group versus doing it to the 2' hydroxyl group first and then transferring it over to the 3' hydroxyl group, but I don't know. I was actually thinking about this reaction today because there's really no reason why, in terms of just the stability, the thermodynamics, there's really no reason why the amino acid should favor being in the 2' or 3' hydroxyl group. You know, it should just be able to isomerize between the two carbons. But in terms of the actual chemistry of when we get to the translation and the actual mechanism of forming polypeptide bond or peptide bonds, it's important that the amino acid is right at that 3' hydroxyl group. So, but yeah, great question. So we have about 10 more minutes, and then we have to have our, where am I? We have our presentations. Okay. Okay, so again, we have a very specific interaction between, so the individual aminoacyl tRNA synthetase, make sure you have that corrected, it's very specific for, you know, both the specific tRNA and then the specific amino acid. And this actual interaction is what we refer to as the second genetic code, and it really reflects this term, the second genetic code, really reflects the fact that you have to have really, really high accuracy of protein synthesis. And part of this accuracy involves making sure that the right tRNA is being attached to the right amino acid. And so there's a lot of proofreading that's involved in this enzyme. So if the wrong tRNA does end up being attached, it will immediately become hydrolyzed. Oh, I forgot. That's what I was going to do today. So I have a handout I'll send to you. I'll actually hand it out on Wednesday. It's a really cool like little reading. But research have been able to generate what's called an expanded genetic code in the laboratory. And this is where tRNAs can be attached to other amino acids other than the common 20 amino acids. So for example, they figured out a way to take a tRNA that's been somewhat modified and attach it to acetylysine. And then, the protein, the protein can then incorporate acetylysine into its overall protein structure, which is pretty cool. So I will get that handout to you guys on Wednesday for you to sort of peruse for your fun reading that you do on the weekends. I think it's pretty cool. Okay, so the ribosome, it is this very complex supramolecular machine. I don't know how super is different from supra. I don't know. Yeah. I don't know if that was meant to be there or not. Okay, so the way that normally this works in terms of teaching is when you start, someone hands you some slides, and then you kind of make it your own. And so someone gave me a set of slides, and I spent a lot of time making it my own. And then I was looking at this, and I'm like, I'm not sure if it really was meant to be supra or not. Okay, so anyways, let's talk about bacterial ribosomes. Amazingly, they are mostly RNA. So it's 65% RNA. So that the salmon. So this is kind of showing the two different subunits of the bacterial ribosome, where you have in salmon, in yellow, it should be DNA, not RNA. And then 35% is protein, so that's your purple, that's protein. So you can see there's clearly much more RNA there. It's actually pretty large in diameter, 18 nanometers. In the context of a cell, that's gigantic. It has a molecular weight of 2.7 million Daltons. So it's composed of these two different subunits that's called 30S and 50S. Those are what's called it's called like a sedimentation coefficient. It's a kind of a way to have a handle on the size of the subunits. And then when combined together, the two subunits form what's called a 70S subunit, or 70S complex. And both subunits have all of these different ribosomal proteins and at least one large RNA, or rRNA. And this is what I think is the coolest part of this. The ribosome is a ribozyme. So it's a catalytic RNA, meaning that it's going to, so the RNA itself is actually catalytic. The RNA is an enzyme, and it's actually, the RNA is involved in forming or catalyzing the peptide bond during translation. So the actual region of the ribosome in which the peptide bond is formed, there's no protein within 18 angstroms of this active site. 18 angstroms is a huge amount of distance, so it has to be RNA that's actually catalyzing the reaction. And so this is just kind of a electron microscope image of a cell. Each of these little dots are, which you can see just thousands of them, those represent ribosomes, so you can actually see them because they're so large. So we're now going to go into the mechanism of translation, that is protein synthesis. And initially you have initiation, elongation, and termination. And so we're going to first go over initiation. So in terms of initiation, protein synthesis always begins at the N-terminal end of a protein, and then it proceeds in the stepwise addition of amino acids to the C-terminal end of the growing polypeptide. So it's always growing from N to C. That's different from, if you guys remember, peptide synthesis in a lab, for those of you who had learned about that. If you do peptide synthesis in a lab, it's in the opposite direction from C to N. The AUG initiation codon is going to specify for the N-terminal methionine residue. So that methionine is always the first amino acid that's encoded in a protein, and that's specified by AUG. So MET has one codon, and all organisms actually have two tRNAs for methionine. So one of those tRNAs is used exclusively when the 5' AUG is the initiation codon for protein synthesis. And then the other tRNA, methionine tRNA, is going to be used for methionine when that methionine is incorporated into an internal position of the protein. So in prokaryotes, there are actually two different tRNAs that are specific for methionine, and they are designated tRNA methionine and then tRNA FMET because the amino acid that's incorporated, so that this tRNA FMET represents that 5' AUG initiation codon, and it will be attached to a methionine that eventually becomes N-formulated. And then just a regular methionine is then used, in eukaryotes, a regular methionine is used at the first amino acid, but there's still a specific tRNA that's used for the initiation codon. And that's different than the tRNA that's then used for internal methionines in the polypeptide chain. And so this is the actual formulation of methionine, of the methionine tRNA molecule. So first, you actually have attachment of the tRNA to the methionine, and then this enzyme called MET tRNA FMET formyl transferase will transfer a formyl group to the amino terminus of the methionine. So that will generate the N-formyl methionine tRNA FMET. And then this is important by having this sort of specific N-formyl methionine tRNA, that sort of is going to help to make sure initiation proceeds correctly. So, the formyl group ensures that the FMET is not incorporated in internal positions of a polypeptide, and that it's bound to a very specific position at in the ribosome during initiation. And so, during initiation, it can only accept this specific tRNA and not the other internal methionine tRNA or other aminoacyl tRNAs. Yes, Marissa.
So with the first part of that, where the first part before it goes to reaction, that's MET TNR FMET, or
Yeah.
so that is still different than from MET TNR and just MET, like on the left side. That is.
This is different than just MET tRNA.
Okay.
Yes. So there's a specific. So what this indicates a specific tRNA specific for methionine that will eventually become formulated. Okay.
I just
Yeah.
wanted to double
Yeah.
check
So
if
the
there's some difference.
so the
Like, it says
tRNA.
FMET on both of them, and I was,
Yeah.
I was like, I'm
Yep.
assuming there's a difference, but I wanted to double check.
Yes. Okay. So let me clarify. So this little F met, the little like superscript indicates that it's a tRNA that is specific for methionine that's formulated. And then the met before that indicates that the tRNA has been attached to a methionine. Okay, and then the N-formyl methionine tRNA indicates that the methionine that's attached to the tRNA specific for F-met has been formulated.
Okay.
Yes.
So I'm trying to think of it in like little chunks. So you start off with the tRNA and it's specifically F-met, and then it will add the met group to the tRNA F-met, and then you will then formulate.
Yes,
that.
yeah. So the tRNA is a specific for its amino acid, which is eventually will be the formulated methionine. All right. So it is 1:21. I'm going to stop this now, and we will begin. We'll talk about the rest of translation on Wednesday, and we will have our presenter.